Jobaline, a multifunctional health formulation developed by Health Forever Products Limited. We will now examine Jobaline's effect on inflammatory reactions involving human cells in vitro. Inflammation plays a key role in numerous pathologies including cardiovascular disease, autoimmune disorders, allergies, metabolic disorders and obesity, arthritis, and neurological disorders. Inflammation may play a role in cancer initiation and progression and plays a role in HIV infection. Inflammation involves a complex system of pathways, including free radical oxidative damage, migration, and infiltration of inflammatory cells into an inflamed tissue, escalating the process by inflammatory enzymes such as COX-2 and pain, among others. Many plant-based compounds can help to block many of these inflammatory pathways. Jobaline was tested for anti-inflammatory properties in several assays using human inflammatory cells. The tests examined whether Jobaline had effects on the production of reactive oxygen species by inflammatory cells and migration of cells in response to the inflammatory mediator leukotriene B4. The graph shows results from treating inflammatory cells with Jobaline before triggering the cells to produce free radicals as part of an inflammatory reaction. Cells treated with Jobaline showed reduced free radical production. This inflammatory effect was also seen for both the water-based and the ethanol extract of Jobaline. This graph shows results from treating inflammatory cells with Jobaline before placing the cells in an environment where an inflammatory mediator sends a signal for the cells to migrate towards the inflamed area. The graph shows the percentage change in migratory activity after cells were treated with Jobaline. It can be seen that Jobaline inhibited the migration. The inhibition was seen both for the water-based and the ethanol extract of Jobaline. The anti-inflammatory effect was seen even at very low doses of Jobaline. To summarize the data shown on the last few slides, Jobaline showed anti-inflammatory properties in several parallel assays using human inflammatory cell types. Please note that both the water-based and the ethanol extract produced this result, indicating that the anti-inflammatory effect is likely caused by multiple compounds acting in synergy. The goal is to advance Jobaline from being seen as a traditional medicinal product to be accepted as a modern nutraceutical product. Science-based support of Jobaline's effects will help gain credibility and increased acceptance of this important bioactive product.